So today let's see the quality control test or the evaluation test for parental preparations. So generally there are three areas for quality control has to be assessed, that is to the incoming stock or in the manufacturing, that is in process quality, and also finished product quality. So the quality control test can be divided into sterility testing, clarity testing or presence of any particulate matter monitoring, then leakage test and pyrogen test. Sterility testing is used to detect the presence of viable organism, microorganism causing of infections in the final preparations, and that must be confirmed with the prescribed pharmacopoeia, and this test is generally performed in aseptic conditions. So the principle is that the product is non-sterile, means they must have some microorganism which is cultured in a nutrient medium, giving the favorable temperature and organism to grow and form colony, and its presence is identified by the means of turbidity. Excuse me. So generally the sterility testing is done by method A and method B. Method A is called membrane filtration method and method B is the direct inoculation method. And the sterility media is also important, that is you can use fluid thioglycolate media or soybean digest cousin medium based on the nature of the organism. So in membrane filtration method, the product can be oily or oily preparations, alcoholic preparations, etc. And if the drug is thermolabile, so here you have a filter, special type of membrane filter paper that is made up of cellulose acetate having pore size of 0.459 micron, and number 2, washing of the membrane by the micro free water or diluting with rinsing the fluid. Or washing the membrane with the product itself 2 to 3 times, then transferring the product into the membrane, that is under vacuum. If it is more viscous, means you have to go for negative pressure application or by using free filled syringes. The number 4 is culturing the broth in the suitable medium and incubated for 14 days. Number 5, interpretation of the result. If you have a microorganism growing, means you can see the turbidity, and if you cannot assess the turbidity, then you further freshly transfer the medium and grow for 4 more days. Then in direct inoculation method, the specific quantity of the product is transferred by means of micro pipette, and of the volume is transferred to the 2 medium each in equal quantity, and it is then cultured, observing the culture during the incubation period. So if the medium is turbid, means further incubated for 4 more days. And if there is no evidence of microbial growth, means the product complies the test for sterility, and if the microbial growth is there, the product is not complies with the test for sterility. So these are the various interpretations. Then clarity test. Clarity is referred to as particulate matter monitoring. So it is either based on visual inspection or electrical method. So the particle can be limited based on the size. So in visual inspection, the filled containers are examined against a strong illuminating background in a swirling motion and looking the particles. Generally you have a white and black background based on the nature of the particle, and this is the visual inspection chamber. And counter method is the test which is based upon the principle that the increase in the impedance or the resistance because of the presence of the particle when it is placed in the two electrode. Similarly, filtration method, light scatter method and light blockage method can also employ. Then leakage test. Here the packaging integrity is tested for hermetically sealed containers for a single dose. If there is a capillary pore, tiny cracks, it may cause leakage, and leakage is intended to detect the incompletely sealed ampules, presence of cracks, openings, etc., so that they can be discarded. So generally performed is a dye test, that is methylene blue is the preferred dye. The under vacuum chamber, it is dipped, all the product are dipped in the chamber which contains a deeply colored dye solution, and the presence of dye inside the container confirms the leakage. So these are the test chamber. Then pyrogen testing. Pyrogen testing is what do you mean by pyrogen? Is pyrogens are the substances present when it is given through ingestions. It will cause the rise in body temperature, and these are generally the metabolic products of the organisms, mostly bacteria, mold, viruses produces pyrogen, and generally they are the endotoxins of gram negative bacteria. So what happens is it will cause the rise in the body temperature. So generally performed by two tests. One is called the rapid test, and then bacterial endotoxin test is also there. So in rabbit test, the organism used are rabbits, healthy mature rabbits. Both the sex can be used, and this procedure generally involves a selection of the animals weighing not less than 1.5 kilograms, and recording their body temperature by using clinical thermometers having precision recording. So prior to the main test, it involves a preliminary test which is called a sham test, where an intravenous injection of the sterile pyrogen free saline solution is given to the marginal ear veins, and the animals which shows a variation in body temperature difference. That is how it is recorded is the initial temperature and the highest recorded temperature, that is the difference. And this reference is, if it is more than 0.5 degrees centigrade, should not be tested in the final. So the main test is performed in groups. First, initially in a group of 3, and here similarly the product is injected to the marginal ear vein. Before it, the product is warmed to the room temperature, that is 37 degrees centigrade, and if it is required, it is to be diluted and injected to the ear vein. The injected volume is about 10 ml per kilogram of body weight of the animal over a period of 10 minutes. Then the recording of the temperature is generally done up to 3 hours after the injection. So the interpretation is that now one should have a body temperature greater than 0.6, and the average of the total temperature sum should not be exceed 1.4 degrees centigrade. Then it passes the test. If it is more than this limit, then the test is repeated using 5 more rabbits, that is the total sample is 8, and the report should be now individual rabbits should have a body temperature of 0.6, and similarly the average should not be 1.4. Then bacterial endotoxin test, it is called LAL test, that is Lemurus amoebocyte lysate test, and this is an in vitro test method. It is more rapid than the rabbit test, and it is semi-quantitative also. Principle is that the endotoxins of gram-negative bacteria in presence of the blood cells of the Limulus polyphemus, that is horseshoe crab, that is called a lysate of amoebocyte, will cause the gelation when incubated at 37 degrees centigrade. And this is the horseshoe crab. This test is also called as bacterial BET test, so can be used as a standardization for endotoxin. So in presence of the endotoxin, the Limulus amoebocyte lysate will cause